Okay, so we're going to continue on with part 3 lecture material, even though we have an exam coming on Wednesday on part 2 material. And on Monday, we're going to dedicate the whole class time to a review session on part 2 material. Okay, so all of the slides that start with systems A, B, C, etc. That's part 3. Again, you're not going to be tested on that until the final exam, whereas all of the organs lectures, organs A, B, C, D, etc., those are fair game for the test that's on Wednesday. Okay, so let's continue with part 3. So we laid out these questions last time. How is energy used? And where is it stored, and how is it controlled? And we talked about the first question mostly last time. So how we use energy, meaning the metabolic pathways that all cells of your body use. And we considered what organs function to control energy, and where in the body we have those storage. Now, on the issue of control, we only just got started before we ran out of time. And we wanted to lay out the parts of a control system having to do with energy in the body. Some folks already knew that the pancreas was a really important organ. And the pancreas features these alpha and beta cells, which function as both sensors and decision-making centers, that is, integration centers for energy balance in the body. So with that in mind, we considered the example of the control of glucose. And I mentioned how the beta cells are excited by high plasma glucose concentrations. And in response to that excitation, they secrete insulin. Okay, so that if on the other hand, you haven't eaten in a while, the beta cells are reduced in their activity. They reduce in their secretion of insulin. Whereas the alpha cells are very excited by that situation. And in response, they secrete glucagon. And so if we look at a time series of glucagon over the course of the day, in response to the ingestion of food and the absorption of nutrients, glucose is our focus, then the glucagon curve kind of looks vaguely like the inverse of the insulin curve, especially around dinner time there where you see insulin go down, the glucagon goes up. And your body pays attention to both. And it's really the relative concentration of insulin and glucagon that have an effect on how the body behaves with regards to glucose that's circulating in the bloodstream. All right, so what are those effects? And that brings us to this question. What are the effects of insulin and glucagon on the cell? So let's step through. We laid out last time how the liver is a big player, skeletal muscle cells are a big deal, and so are the adipose cells that are in the fat stores in your body. So let's step through and consider how those cells are affected by insulin and glucagon. Okay, so here we have a hepatocyte, a liver cell up top. And we're going to sort of keep the control stuff down in the bottom, but then we're going to see up above what's happening at the membrane of these particular cell types. So for the liver cell that binds insulin, okay, so let's think about this. So I eat a meal and insulin goes up. So all of a sudden, when you absorb those nutrients, your bloodstream is going to be rich with nutrients, insulin in particular. Glucose in particular. And you want to have that source of energy for later in the day when you haven't eaten in a while. And so the goal here is to store it so that it can be released later on. All right, so all of these steps, just keep in mind what the goal is. So the goal is, again, to store energy for later. All right, so insulin is the signal to do that. So when this hepatocyte binds the insulin, then it causes a sequence of changes inside. Now, here's an example of a second messenger pathway. And our treatment of second messenger pathways is very much black boxy. Okay, we have an arrow that where insulin triggers the second messenger pathway, and then through mysterious means involving transcription, enzymes, various things that happen inside of the cell, the hepatocyte will take the glucose and store it in a different form. It'll transform that molecule so that it is more stable for storage. And in particular, it takes the glucose and it produces glycogen from the glucose. Now, not only, and a cell's ability to store energy as glycogen is limited. That's sort of like you have a certain number of shelves in the cell, and then they get stocked up. And once they get stocked up, then you want to take the remaining glucose and store it away in long-term storage. And the long-term storage is in the form of triglycerides. So we mentioned that we have glycogenesis and lipogenesis in those pathways where we had sort of buckets of energy in the body. And so those terms are just being introduced again. Another thing a hepatocyte will do, again, that's mediated by these second messenger pathways, is they'll burn up glucose. So we have, again, in the cytosol of a cell, the ability to produce a couple of ATP for every glucose molecule through glycolysis. And so glycolysis goes up. And basically, that's just going to be good for that one hepatocyte because those ATP produced there are going to be of no use to the rest of the body. But at least it reduces the ATP that's in the bloodstream. Okay, so those are all the pathways for the liver cell. All right, it looks a little ugly, but you're going to see very similar boxes and arrows for the other two cell types, and there's only 3 cell types that we have to worry about. All right, so we're going to take that response and plug it in. So since it's a response to this stimulus, again, stimulus was the concentration of glucose. We're in the efferent domain, that is things that are done in response to the stimulus, which we're going to show in red. So when liver cells get that insulin signal, they do these pathways, which we see illustrated up above. So we're going to plug that into our control system. Okay, now that's what happens right after you eat a meal. Now what about when you haven't eaten a meal in a while and you'd like to cash in some of the energy that you stored away so that it is in your bloodstream, there is a decent amount of glucose that's allowed. Okay, so that's mediated by glucagon. Again, the alpha cells do that. And so we're going to stick with the hepatocyte up above. Now we have glucagon circulating around, and glucagon binds to a receptor. We've got another second messenger pathway that is stimulated by the binding of that glucagon. And here we're dealing with, well, in the case of glycogen, the same molecule, but we want to go in the opposite direction. We want to cash in that glucagon for glucose. So glycogenolysis is just that. It's reversing the glycogenesis that we had in the hepatocyte when it bound insulin. Now, hepatocytes also have this sort of superpower of being able to take proteins, and in particular amino acids, and producing glucose from those as well. So if you really haven't eaten in a while, then it will start to do that. So gluconeogenesis, or new glucose from amino acids, will also be stimulated. Again, it's all triggered by that glucagon that's circulating around. So with these glucose that are being essentially liberated from their long-term storage inside of the cell, once that rises up in the interior of the cell, then they can move back into the bloodstream. Okay, so again, homeostasis, you always want to try to maintain your state variable at a constant level over time. So that state variable here is the concentration of glucose in the bloodstream. You get a big surge, and homeostasis should try to counter that surge when you eat. And then when you haven't eaten in a while, then it should be liberating those stores in order to try to raise it up, even though you haven't gotten anything from your food in a long time. Okay, so we add those to our efferent pathways, glycogenolysis, glyconeogenesis. Okay, what are the cell types? Well, we've got the fat cells, the adipose tissue, and resting muscles. Okay, so everything I'm going to show above, you can imagine it's either a resting muscle or it's a fat cell. Both do the same thing in response to insulin. So the insulin binds the receptor, again, a magical second messenger pathway. In this case, the second messenger pathway will facilitate the absorption of glucose so that it enters inside the cell. We have glycolysis occur. That is just the burning up of glucose so that those cells have more ATP. All right, so we add those. The adipose tissue or resting muscle cells are added as efferent responses. For just the muscle cells and not the fat cells, we also have the ability to produce glycogen. So adipose cells do not produce glycogen, and so just like a hepatocyte, a resting muscle cell can take some of that glucose and store it away into glycogen. Again, short-term storage. And so we add for the muscle cell at rest what we have for the adipose tissue cell plus the glycogenesis, the production of glycogen, and then there's also, which is not shown here, protein synthesis. That's also a response to insulin secretion. Okay, so that maps out a whole bunch of pathways in response to insulin and glucagon in these cell types. So if we consider the plasma glucose, again, is the stimulus. Let's consider what happens right after a meal with all of these details added. Okay, so after a meal, your body is in what's called the fed state. Okay, so in the fed state, you have a rise in plasma glucose. And so we've already mentioned how that rise is going to stimulate the beta cells. This is nothing new. But all of these pathways are going to result in the transport of glucose from the blood into liver, fat cells, and muscle. And then the conversion of glucose, either burning it up into ATP via glycolysis or all these other ways that we just itemized to then deal with the glucose that's inside of the cell. All of those are going to work to reduce the concentration of glucose that's circulating around in the blood. So it has a negative effect. All right, again, this is right after you've eaten. The alpha cells, their job is to deal with what happens after you haven't eaten in a long time. So they are inhibited at this time. And so all of those efferent actions that you see under the alpha cell diamond, all of that stuff is going to be inhibited. And as a consequence of less of all of that stuff, then it too has a negative effect on plasma glucose. So in other words, so the ability that the liver has to produce glucose from proteins, that whole process is inhibited when plasma glucose is high. So the alpha cells reduce their glucagon secretion, and as a consequence, you get less of that. Okay, so they're not going to be releasing, those cells will not, or that pathway anyway, will not be producing more glucose that is then going to be added to the bloodstream because we just ate a meal and we don't want that coming from the liver. Now, if you haven't been eating in a while, we have everything essentially reversed. Okay, so the stimulus here is low plasma glucose, so that goes down, which has an inhibitory effect on the beta cells. And that in turn negatively influences plasma glucose. So all this stuff under the beta cells is about storage. And if we haven't eaten in a while, we don't want to be activating storage. Okay, and so if we inhibit the creation of storage, or the storage of glucose, then we negatively will influence the plasma glucose. And if the alpha cells respond positively by releasing glucagon, then we have a liberation of glucose and then a release of glucose into the bloodstream. Again, all in the interest is trying to keep it stable over a long period of time. Any questions? Yes?
So what I understand is what you said, Professor, is if you just ate, you're going to want to lower the glucagon signal, right? And if you haven't ate, you want to lower the insulin signal, but then increase the glucagon signal. Is that right?
That's correct.
Okay.
Yeah.
But what are you using for gluconeogenesis and glyconeogenesis? I can't say it. If you haven't ate.
You're talking about these ones on the right under the alpha cells?
Yeah.
Yeah.
Because I read somewhere that you could use the amino acid from like proteins for glyconeogenesis. Is that true?
Yeah, so the liver cells are capable of producing glucose from amino acids. So that's gluconeogenesis.
Yeah.
But they only do that when it makes sense to do that. And it only makes sense to do that if you haven't eaten in a while.
So they do break down proteins in the body for that?
Yes, they do break down proteins, and then ultimately this pathway is taking amino acids from that breakdown and producing glucose. But again, you'd only expect that when glucagon is at a high concentration in the bloodstream.
Thank you.
Yeah, question?
For the fasted state and fed state, is that like a pretty standard period of time, or is that relative to each person?
Oh, so the question has to do with sort of the time scale of the fasted state and the fed state. And I would say that, yeah, to some extent, it's going to vary from individual to individual. And it also varies with what you've eaten. Your feeling of satiety varies with your diet. This is one argument for eating fat because it stimulates satiety. So you feel like you're satiated or that you're fed for a longer period of time when you've eaten a fatty meal. And fat is more energy dense, and so it kind of makes sense that your body recognizes that I have just eaten a fatty meal and I'm going to be okay for a longer period of time than, say, a carbohydrate-rich meal. So it can vary with diet, and it can vary with exercise, and it certainly varies with metabolic rate. And we all have different metabolisms, and that varies a whole lot depending on a variety of factors. So yeah, it's all subject to lots of sources of variation. Yeah. Okay, so let me pose a scenario. Given this control, here's a scenario. Type 1 diabetes. Now, we don't talk a lot about disease states or diseases, in part because I'm just trying to explain how things work in a healthy state. And once you involve diseases, sometimes things get even more complicated. But here's an example of a disease that I think is beneficial for understanding how things work in a healthy condition. So type 1 diabetes is an autoimmune disease where your body starts to destroy beta cells. So beta cells, okay, so they secrete insulin in response to an elevation in plasma glucose. So you're effectively removing them from this control system. Okay, so as a consequence, you're eliminating that negative feedback that is provided by the beta cells. That is our homeostatic response when we eat a meal to keep plasma glucose relatively modest. And that means we have this situation of hyperglycemia. And hyperglycemia damages the kidneys, the eyes, your nervous system. Glucose might taste good, but it can be damaging at really high concentrations. So it makes sense that a solution to type 1 diabetes is to take the place of beta cells by manually injecting insulin. And so when you do that, you rescue these efferent responses, all of those responses that we itemized, to insulin. Those are achieved by injection. And so, as you might imagine, being able to inject just the right concentration under just the right circumstances is a little difficult, and it can only really be estimated because the beta cells, when they're healthy, are constantly monitoring plasma glucose and making these adjustments of insulin concentration. But at any rate, we're essentially returning to a situation where you have negative feedback in response to a surge of glucose. All right, so that brings us to a clicker question. Okay, so the clicker question is, so what I just described is type 1 diabetes, okay, which is that you're effectively removing the beta cells. There's a different type of diabetes, type 2 diabetes. And here are the symptoms. Type 2 diabetes includes elevated concentrations of glucose, but also elevated insulin in the blood. You wouldn't expect that for type 1 diabetes because you don't have the beta cells. So what is the likely cause of type 2 diabetes? Is it A, insensitivity of beta cells to the concentration of glucose? B, insensitivity of cells to insulin? C, increased glucagon secretion? D, insensitivity of systemic cells to glucagon? Or E is liver failure? So take a minute to think about that. Okay, give me your final answer to the question. Okay, 3/4 of you got the right answer, which is B. All right, so type 2 diabetes makes cells less sensitive to insulin. So where it's acting is not on the beta cells like type 1. Instead, you just have a reduction in all of these efferent pathways because the cells become less sensitive. And so this is associated with diet, high sugary diets, a lack of exercise, obesity. These are all contributing factors to type 2 diabetes. And what this illustrates is essentially how it acts. And so that's how you get, if your body is less sensitive, then what the beta cells start doing is ramping up the insulin that's secreted so that you have high insulin and high glucose. The cells are less responsive, and the beta cells are clever enough to then therefore try to get some sensitivity by increasing the insulin concentration. So the variety is a big area of research to get your cells responsive, but being active and adjusting your diet are big treatments for type 2 diabetes. Okay, so that's the end of the energy balance stuff. So I'm going to switch my slides. I just need a minute for that.